Hello, my name is Teemu Miettinen. I am a cell biologist and I work here in the Koch Institute as a research scientist. And today I'm going to tell you about how we have taken our advancements in developing tools for cancer research and also applied those same tools for the study of marine phytoplankton. Now, what are phytoplankton? Phytoplankton are single-celled and multicellular species that inhabit all of our oceans. They are essentially plants of the ocean, so they are responsible for carrying out approximately half of all the global carbon fixation that takes place on our planet. They are understudied and highly diverse, which means that they are genetically very diverse and they are also morphologically very diverse. And in our work, we focus on single-cell phytoplankton, such as the one shown here in the image. And while these species are largely understudied and we don't know tons about many of them, we do know that most of them are phototrophic. And what that means is that these species get their energy from sunlight. And this means that the cells must balance the depth within which they are in the ocean water column. So the cells want to get enough sunlight and be close enough to the surface of the ocean to get that light, but the cells also need nutrients, the different kind of building blocks that they use to make stuff. And those are more abundant deeper down in the ocean. So depending on the environment, the cells might want to move up or down to find more lights or more nutrients. And this is essentially what we study. We want to understand how these single-cell species are changing their gravitational sinking in response to changing environments and conditions. Now, studying something like this is surprisingly challenging because how do you measure the gravitational sinking of a single cell? Luckily, the laboratory of Professor Scott Manalis here in the Koch Institute has developed a tool called Suspended Microchannel Resonator. Now, this tool is essentially a scale that allows you to weigh how much mass a single cell has. Now, I came here to the lab of Professor Scott Manalis already 9 years ago, and it was heavily driven because they had this tool, because it's an extremely good tool to study different biological questions. And since those days, we've jointly developed volume measurements on top of these mass measurements, so that when we weigh a single cell, we can get both its mass and volume. And of course, if you know the mass and volume, you can solve for density. And with that information, you can calculate the exact gravitational sinking velocity of a single particle. So we have a way to actually determine how fast single cells are sinking. And we can do very simple experiments. We can just take our phytoplankton and put them into different kind of nutrient environments. And what we see with many phytoplankton is quite surprising. The phytoplankton seem to sink more when they are starving for nutrients. And this is not at all obvious because if you just image these cells, you don't see a difference. The volume is basically the same whether the cells are in high or low nutrient environments. But the reason they sink more is that there's more mass and more density. And we believe that this higher sinking is the cell's adaptation to sink deeper in the ocean to find the more nutrient-rich waters. Now, this may not seem that surprising to you, but let me put this in different terms. When phytoplankton go on a diet, they gain mass. And that is not at all obvious at first sight. But the reason we can discover things like this in the first place is the fact that we have these measurement tools. And these tools were originally developed for cancer research, and they have been very powerful in studying cancer in different contexts. But with good, high-quality basic method development, we can apply those methods to a variety of fields. And the reason we also want to apply these methods to things like marine microbiology is that it's simple organisms, such as this phytoplankton that we have studied, that have a large role in supporting marine ecosystems and the climate of our planet. And if we can only understand the physical properties of these cells better, then we can start understanding how these cells and these species are responding to changing environments and conditions that are on our planet. Now, with that, I would like to thank members of the Scott Manelson's lab. We have had excellent postdocs and students helping us in this work. We've also had excellent collaborators from the lab of Martin Boltz. And these beautiful images that you see are only possible because we have core facilities here at MIT that support us and help us capture images like this. And last but not least, I would like to acknowledge our great funders who support both of our environmental studies and our cancer research. Thank you.